Hey students, welcome back to class. Today we are going to be talking about the stages of protein synthesis, transcription, and then translation. The section about splicing and processing is kind of an intermediate. It's something that does, in fact, of course, happen, but it's not one of the main stages. The main stages are transcription and translation, and we're also going to be looking at the mechanism of those stages and the regulators for those stages. So how do we keep transcription and translation at the proper amount so that we're controlling the amount of gene product that we're making? So we're going to start out by looking at transcription, and that word transcript should help you. A transcript is a copy. So if you were in a meeting and someone's taking notes about the meeting, that would be the transcript of the meeting. They're writing down what was there. You're not in the meeting, but you get to hear about the meeting and understand the meeting because you have a copy of what happened in the meeting. So in transcription, we are making that transcript. The mechanism for making the transcript should feel very similar to the mechanism for replicating DNA. We're going to see helicase come in. We're going to see the transcription start, but this time it's going to be RNA polymerase that's going to show up and start transcribing. And because we only need to make a single-stranded piece of RNA, instead of having a leading strand and a lagging strand, we're just going to show up and on the 3', so you can see the little 3' here, and so we follow that along. That's where our transcription is going to start, and we've got this big blue RNA polymerase behind, and it's just going to open up the DNA and start transcribing in its 5' to 3' direction, opposite to the 3' to 5'. It's just going to go, and we're going to end up with this 5' end as the origin, and it's just going to keep transcribing until it reaches the end of the gene. So in each chromosome, there are these genes, and at the beginning of the gene, there is something called a promoter, and that's where the transcription is going to start. And at the end of the gene, there's something called a terminator, and that's where the transcription is going to end. So during transcription, we take down a copy of all of the nucleotides, the A's, the U's, the G's, and the C's, because remember, there's no thymine in RNA. We take down all of the nucleotides from promoter to terminator. And then that product we now call the mRNA. That product gets ready to go out into the cell and be translated into a protein. So we mentioned that as we discussed each of these stages, we would also talk about the regulators. So we said before that RNA polymerase needed to come in and bind to the promoter. And I'm just going to kind of up the level a little bit on the details of that interaction. So if you'll just look with me for a second at this picture. In blue, we have still RNA polymerase, still in that light blue, just like it was on the previous picture. And we have this section of the gene that we call the promoter. But within the promoter, at the very beginning of the promoter, there is a place called the TATA box, or the TATA box. And that's repeats, DNA repeats of thymine, adenine, and it makes this TATA box. The TATA box helps to call over the RNA polymerase in order to start getting transcription. And then once the RNA polymerase is there and we get some of our what we call general transcription factors, we can get other regulatory proteins that help kind of really shove the RNA polymerase off and down to transcribe. So I don't know if you guys have ever seen Cool Runnings, but Cool Runnings is a movie that's about a Jamaican bobsled team. And that's the only time I've ever really watched bobsledding. But when I look at this diagram of RNA polymerase about to transcribe a gene, I kind of see the beginning of a bobsled race, right? So if you've got RNA polymerase and it's the bobsled, right? But in order to get the bobsled to move, if I just leave it up there at the top of the slalom, it's not going to go. You have to push it to start, right? And so as we think about pushing it to start, that's what we need these transcription factors for. So all these little circles, the orange and brown, pink, green, and gray circles that are here, those are kind of like the bobsled team members. And they've really got to push off this RNA polymerase in order to get transcription. These other transcription factors that are here help even give it a bigger shove so that it really gets going. If you ever tried to bobsled, you might realize if you start running the bobsled forward, you might not get enough speed to make it all the way down the track. You would stop midway. You need a really solid shove at the beginning. And so all of these other transcription factors are helping to make sure that we get that shove. When we talk about the system in terms of regulation, you always have the TATA box and the promoter here. You could always get RNA polymerase to come over. But in order to really get that push, you need all of these different proteins. So imagine to get this one gene product of this red gene, you need these three yellow plus 2 blue, that's 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. In this drawing, it's showing 18 different proteins that also need to be in the right place at the right time, in the right cell, on the right gene in order to get transcription. And so by making it so complicated to start it, you make very sure that you never accidentally make a gene when you didn't mean to. You never accidentally make its product. You never accidentally start producing hydrochloric acid on your skin cells or growing hair in your esophagus because you've shut those genes down. They don't have the regulatory proteins that they would need. And so this is one way to regulate it. If you don't have the right ingredients, you can't make the mRNA in order to go make the gene. In addition to having the right proteins there, they also need access to the TATA box and the promoter. And so we're going to look at what happens, how we can keep them from getting access to those things. So I'm just going to do one slide on this process called epigenetics, but I want you to know that epigenetics is something that we could talk about for days. Epigenetic issues, epigenetic causes are probably responsible for more illnesses, chronic illnesses like type 1 diabetes or even cancer than they have previously had credit for. And epigenetics is an emerging section of biology that is just not only fascinating, but very important to your future health. And so I'm going to try to give you a really solid foundation on it, but I'm really going to encourage you to take the time to go look up some things on epigenetics. And actually, right after spring break, I'm hoping we're going to have the opportunity to take just a really quick look at epigenetics because I think it's such an interesting and emerging portion of biology. This starts with those tightly packed histones and our packed up DNA. Remember we talked about DNA when it's going on a journey, when it's traveling, it wants to really pack up tight, but then oftentimes once it gets to its destination, it gets put into its cell. It just kind of leaves its DNA kind of loosely packed. Well, epigenetics looks at how we pack that DNA. So the first thing we need to look at is histone acetylation. So an acetyl group are these little yellow dots, and an acetyl group is a chemical group. And those groups can be added on to histones. And so as we add the acetyl groups onto histones, the histones move away from one another, loosening and unpacking. And so what this allows us to do is to get access to the DNA that's in here. So when it's time to transcribe a gene, before we can get the RNA polymerase at the promoter, before we can start to make this template of mRNA, we need to loosen the DNA. We need to spread the DNA strands out so that the RNA polymerase can come in and have access to them. The histones, those are the blue proteins that are here. The histones can get acetylated to spread out the DNA. But the DNA itself can be methylated. Methyl is another chemical group. And it's not just a chemical group, it's kind of like a big branching chemical group. It's large. And so when methyl groups become attached to the DNA can't get transcribed because the methyl groups are already in the way. If you've ever seen 101 Dalmatians, there's a part where all the kids, all the little puppies are trying to nurse, and Rolly is trying to get in there and he like can't get in because everybody's in the way already. That's what I think about with DNA methylation. It's like all of the methyl groups are in the way. So even though the promoter's there and the transcription factors are there and RNA polymerase is there, they can't actually get through the door to get to the DNA to do the transcription. They end up stuck outside. The really interesting part, and the reason why this becomes a whole nother branch to study, is that the methylation of your genes get passed on from generation to generation. It does not get wiped clean with a new baby. So if your DNA for a particular cancer-causing gene has become unmethylated or demethylated, so you don't have the methyls on it, so the gene is getting transcribed, and that is causing cancer, that tendency gets passed along to the next generation. Likewise, if you have, let's say, BRCA gene for breast cancer, but that gene is methylated, the gene stays off for your lifetime, and those methylations get passed along to the next generation as well, keeping your offspring safe. So this leads us to this entire concept of called epigenetics. Epigenetics means above the genes. So above whether or not you have the gene for breast cancer, you talk about the controls of that gene for breast cancer. And so the idea is that we might be able to prevent something that has a genetic cause, like breast cancer or schizophrenia, by identifying the gene and methylating it, blocking it from being transcribed, blocking it from being expressed. We can prevent that gene from being expressed and potentially save the lives of people with those genes. So this is one way where the study of protein synthesis and epigenetics and genetic controls impacts your life and health. This is something you might read about in the news. This is something you might hear about in the treatment and prevention of diseases like cancer and schizophrenia and even Alzheimer's and heart disease. So all of those things I just listed have genetic basis. And if you can switch off those genes with your DNA methylation, then you can increase quality and length of life. So quick recap, in order to get gene expression, we want lots of histone acetylation and very little DNA methylation. You should write that down. In order to get gene expression, we want lots of histone acetylation and very little DNA methylation. In order to turn off a gene or turn down or inhibit a gene from being produced, we want no histone acetylation. That would make the gene tightly packed. And we want lots of DNA methylation that would block the gene. So to turn down or off, no acetylation, but lots of methylation. Those terms might be difficult to keep track of, so I would really suggest writing down those relationships in your notes. Once the DNA transcript is made, it's time to splice and process it. Splicing and processing are getting that gene product ready for translation. Splicing is where we remove something called an intron. An intron is an interruption, or more appropriately, a non-coding portion of the DNA. The introns are important. They do things like regulate genes in other parts of the DNA chromosome, but the introns are not needed or a part of the genetic code for that particular protein. So we need to splice them out. You can see the introns getting moved down here in this gene. So we have our 5' end over here and our 3' end. We have exon 1, intron, exon 2, intron, exon 3. And what we would do here is we would splice out the introns. I do that when I make these videos. Sometimes there's some dead air. And so when I go back through, I splice out those quiet portions or portions where I mess up a word so that what you watch is just a smooth, completed video. That's what the RNA does as well. So it puts exon 1 next to exon 2 next to exon 3. That's the splicing. It also does something called processing. Once the mRNA moves into the cytoplasm, there are many enzymes and other things out there that might harm it or break it down. So the mRNA gets processed. It gets a guanine cap on the 5' end, and this helps the ribosome to find the portion of mRNA that needs to be transcribed. And it gets a poly-A tail. And so the poly-A tail is where the digestive enzymes would be chomping down the gene. But we don't want them to chomp it down before we get it translated. So we put this kind of buffer of these poly-A's on the tail so that as those digestive enzymes come through, what they're chomping on is something that's not necessary for the gene product. So we put a cap to help it find where it's going and a tail to help protect it from breaking down. We get rid of the introns and we put all the important things together. So that's our processing. We mentioned again that we can regulate at really every portion of this process. So in this case, we find something called alternative splicing. What that means, think of reality TV. When you watch reality TV, I watch Top Chef. I used to watch Top Chef all the time. And a long time ago, there was a character on Top Chef named Stefan. And he was totally the villain of the season. Nobody liked Stefan. He was so mean and arrogant and rude and caused all sorts of problems. And it just so happens that after Top Chef ended, Stefan opened a restaurant in LA. And my husband and I went to go eat there because anytime a Top Chef opens a restaurant around us, we go eat there. We went to Stefan's restaurant and we talked to him, and he was so nice and so kind. And we said, Stefan, on Top Chef, you seem like such a mean guy. And he laughed and he was like, editing. You know, because the editors could take an eye roll that Stefan did 3 weeks ago, and they could splice it in right after one of the beloved characters says something naive, and then they show Stefan rolling his eyes. And so they're making a narrative. They're making the story different than it actually happened by the way they edit it. mRNA splicing works the same way. So if you look at this piece of mRNA, okay, so this is our DNA up here, this is our primary mRNA down here. We show 3 different ways that we could splice this one piece of mRNA to make it into 3 different proteins. In my first mRNA, I remove all of the introns and exon 3. So see, it goes 1, 2, 4, 5, 6. In my second protein, I remove exon 2 and 4, and so that makes a different protein. And in my last protein, protein 3, I remove exon 2, and so that makes yet a third protein from this one piece of mRNA. This creates an exception to what scientists believed was true, that for every one gene there was one protein. Now we're saying that one gene makes just a small handful of different proteins. And so it might be true that protein 1 is how we would splice it if we were in the brain. But protein 2 is how we would splice it if we were in the spinal cord. And protein 3 might be how we would splice it if we were in the muscle tissue. So different cells may splice the same gene different ways to make different proteins. So this is another way that we can control protein synthesis. We can control it with our DNA methylation, our histone acetylation, and now with alternative splicing, all ways that we can control which gene products are being made. So taking a second to just look back at where we've come from, we have managed to talk about transcription, where we change our DNA into mRNA. We have identified that that's regulated by the packing of the histones with our acetylation of the histones and the methylation of the DNA, and by the transcription factors, whether or not they're present. We've also talked about the splicing and processing, the preparation of the mRNA to leave the nucleus, and that can be regulated by alternative splicing. When we meet tomorrow, we are going to discuss the mechanism of translation. How do we go from our mRNA transcript to a functional protein that acts in your body? We'll be refreshing our stages of protein synthesis and also looking at mutations when we get together in our next video. Good job today, you guys, and I will see you in class.